Welcome to the Silicon Valley Health Institute. I'm Susan Downs, and this gentleman on the screen as well is Stephen Fox.
Howdy.
Hi all. And with us we have the pleasure and luck of having Doris Loh. She is a researcher who has been looking into current situations and coming up with some very helpful information that will help us. She's trained as a classical pianist and has done extensive research on ascorbic acid and melatonin. She is focused on how health and disease are greatly affected by electromagnetic radiation that surrounds us. It's everywhere we go. Her work on EMF offers insight and solutions into the challenges and other living organisms are facing during this era of change. And believe us, the EMF is coming all around us. Other major works that she's focused on are on deuterium, as well as a startling series on the quantum features of the major redox balancer vitamin C, that's ascorbic acid. Her ongoing series on the current virus that's challenging us all is recognized around the world for its in-depth coverage of this disease and discussions in the holistic alternatives that will help us get well, build our immunity, help prevent, and help get through it. So welcome, Doris.
Thank you, Susan, and thank you, Steve, for having me on the show today. So I have a lot to present to everyone, and I think we'll just get started. Today's presentation is called Ancient Molecules for Healthy Hearts, and it's going to focus on a really major aspect of how the virus SARS-CoV-2 affects humans, and it's not being paid attention to. And so let's start by taking a look at what's going on with the virus infection. As of a few days ago, we have 32 million people being infected. And daily new cases are still up there, and death rates have come down a little bit, but it's still being steady. And a lot of people have said a long time ago when the virus started that by summer or warm weather, the virus is going to slow down. Well, it hasn't. And a recent study using a math model to correct biases due to incomplete testing and flaws in testing accuracy actually has estimated that there, between the period of February 28 to April 18, there's actually at least 3 to 20 times higher confirmed number of infected than the confirmed cases being reported. So could be a substantial underestimation of how many people are actually infected by the virus in the United States. And from a global survey of 640 patients from around the world who reported their symptoms, there is about 62 COVID symptoms being reported over a period of 8 weeks. And the most interesting and concerning symptom that I found is tachycardia, or rapid heartbeats and palpitations, because they were reported in over 37% of the respondents in this survey during that 8-week period. What most people don't realize is even though the infection rates have gone up, and most people are very happy to say that the death rates have actually come down, but there is one aspect that people are not paying attention to is that COVID-19 patients may be at risk for cardiac dysfunctions. Cardiovascular disorders, including myocardial injury, arrhythmias, acute coronary symptoms, and venous thromboembolism has been reported. And higher mortality rates are actually associated with COVID-19 patients with underlying cardiovascular conditions. And in general, left ventricular diastolic and right ventricular functions are impaired. In hospitalized patients, the patterns of cardiac disease is like this. A major number of the patients, 39% of them, have right ventricular dilatation dysfunctions. 39. This is a large part of cardiac disease being reported in COVID-19 patients. Right ventricular dilatation. Now I want you to remember this. And then people who have recovered from COVID-19, they actually develop sustained cardiac involvement. 78% of patients show cardiac involvement in their MRIs taken 71 days on average after recovery. And 60% of those patients have ongoing myocardial inflammation. And that is independent of pre-existing conditions, severity, overall course of illness, and the time from the original diagnosis. And yet another study found sustained cardiac involvement in 58% of patients recovered from COVID-19, where their major cardiac magnetic resonance presentations were, again, impaired right ventricular contractile functions. Now in children, I'm sure you've remembered a few months ago that there were children being presented with symptoms similar to Kawasaki disease. Now this type of symptoms is being called multisystem inflammatory syndrome in children, MIS-C. A systematic review of 662 MIS-C patients found that 54% of them had abnormal echocardiogram and 31% had abnormal EKG. And then another study that reviewed 8 documentations found that 440 MIS-C cases had cardiovascular symptoms in 71% of the patients. Now, the strongest predictor of myocardial injury in MIS-C is, again, right ventricular strain. Now, even though there are usually signs of recovery of systolic function during the subacute periods, the diastolic dysfunctions persisted. Now, I want you to remember right ventricular strain and diastolic dysfunction. Now, MIS-C patients, the children, even if they had preserved ejection fraction, they all seem to have exhibited subtle changes in myocardial deformation that may be a possible indication of subclinical myocardial injury. Now, there is actually evidence of direct viral infection of the heart. A recent report on 39 autopsy cases of COVID-19 patients with pneumonia as clinical cause of death, 61.5% of them had viral particles in the heart. And there was also evidence of active viral replication. Now, cardiomyopathy and heart failure that's related to COVID-19 may actually, in my mind, potentially become part of the evolving story of COVID-19. So how does SARS-CoV-2 infect cells? Now, this is a diagram of the SARS-CoV-2 structural proteins. there are four main proteins. The spike protein, the S protein, this is the spike. The N protein, right here. The membrane protein, what they call M protein, here. And the envelope protein. S, M, and E are part of the envelope, while the N is a protein bound to the viral RNA in the core. And the spike protein, what they call S protein, is actually responsible for both binding to receptors on the host cells and membrane fusion events. So binding and fusion are two processes that the virus uses to invade cells. Now, SARS-CoV-2 is what is known as a class 1 coronavirus. There are 3 classes, but SARS-CoV-2 is class 1. Now, to infect cells, these envelope viruses, and all coronaviruses are envelope viruses, they must fuse with the cell membrane. And they have dedicated fusion proteins that they use to fuse with the cell membrane. Now, before the fusion proteins are activated, they have to be primed by a proteolytic agent, cleavage agent, that converts them into fusion competent states to prepare for that fusion event. And for that to happen, you actually need another trigger. And in SARS-CoV-2, the triggers for that fusion is receptor binding followed by protease action. Now this, remember the spike protein I showed you just now? This is the spike. And this is a close-up of that spike. Spike protein, and this is what it looks like. It actually has 2 subunits. This is subunit 1 and subunit 2. Now the spike protein subunit 1 and subunit 2 must be cleaved. That means they must be chopped up. Subunit domains. The subunit S1 has the NTD, N-terminal domain, and the receptor binding domain. And S2 consists of fusion peptides, FP, HR1. And before you can have receptor binding, fusion, and cell entry, these 2 subunits have to be cleaved, meaning separated. Now, the interesting thing is, pre-cleavage processes could differ by cell types. In human airway epithelial and lung cells, SARS-CoV-2 virus must be pre-cleaved by the furin protease before the spike protein can be further activated by proteases such as TMPRSS2 and cathepsin B and L. Now, it is believed that the pre-cleavage by furin decreases the overall thermal stability of the virus and therefore facilitates the adoption of an open conformation that is required for binding to receptors such as ACE2. However, now remember I said pre-cleavage is kind of different in different cells because what they found in an experiment involving stem cell derived cardiomyocytes is when they inhibit furin, they did not reduce viral infection. Now this is a furin inhibitor. This shows the amount of viral proteins in the cell. This is the control. When they inhibited the ACE2 and a cathepsin, the viral proteins are decreased. But when they inhibited furin, it didn't do anything. Now, the SARS-CoV-2 has a cleavage site for furin, and that makes it actually different from SARS-CoV-1, which doesn't have furin cleavage sites. And SARS-CoV-2 actually has 3 furin cleavage sites with different functions. This is F1, F2, and F3. F1 can promote fusion events. F2 participates in pathogenicity after the cell entry. This is F2. And F3, which is in the receptor binding domain right here. Remember S1 receptor binding domain? They promote adhesion between the virus and the cell surface. 3 cleavage sites. 1, 2, 3. Now, what makes this furin cleavage site so important is, yes, it needs furin to be cleaved, but it doesn't have to be furin, depending on the cell type. And in cardiomyocytes, and other types of cells, they found that the SARS-CoV-2 can be cleaved by several, 1, 2, 3, 4, 5, 6 types of proteases. And they compared the cleavage to SARS-CoV-1. The blue is SARS-CoV-1 and the red is SARS-CoV-2. Furin, this is prohormone convertase, and trypsin, maltryptase, cathepsin, and cathepsin L. The activation of the spike protein is actually a complex process involving multiple cleavage events. And remember, I just showed you there are 3 cleavage sites. So multiple cleavage events at distinct sites with different host proteases. So any of these proteases can be involved at any time. And SARS-CoV-2 is different from SARS-CoV-1 in that it has a unique 4 amino acid insert in the S protein at the S1 and S2 site. And this site allows the SARS-CoV-2 to be cleaved by a wide range of proteases, including furin. And once it's cleaved, it can be followed by receptor binding. Now, with receptor binding, it doesn't happen automatically. The receptor binding domain in the spike protein can either be closed or open. Now, when the RBD is in a down or closed conformation, ACE2 or other receptors cannot bind to the spike protein, whereas when the RBD is an up or open conformation, then that domain is accessible to receptors like ACE2. And switching from a down to an up conformation for the receptor binding domain in the S1 subunit is a prerequisite for binding. Now, this is the receptor binding domain in the down position, and this is the up position. And this is a close-up of that. Now, when it is up, you can see ACE2 being able to bind to it. Now, why am I talking about the open and closed conformation? Well, because the SARS-CoV-2 virus has mutated. There is a major mutation or variation that took place in the important spike protein, where a key amino acid changed from aspartic acid, which is D, to glycine, G, at position 614. That's why you see people referring to the D614 virus and the G614 virus. This G-clade mutation was very rare in January. You don't see any. This is January. But it gained global dominance starting in Europe, beginning around February. See, around the end of February, it's half and half, and by now, it's mostly the G-clade. And what's up with the G-clade? Now, this is the D, and this is the G. Look at how it is able to infect cells. The D-clade didn't infect too many cells, whereas G-clade can infect a lot of cells. The 614G variant is now the most prevalent form globally. Scientists believe it harbors a fitness advantage and immunological resistance. So infected individuals actually show higher viral loads in their upper respiratory tracts that is associated with lower viral cycle thresholds. Now, the good thing maybe is that the G variant is not associated with increased disease severity, but it does have greater infectivity due to increased fitness. And the key amino acid changes in the spike protein actually influences how the virus infects human cells. Now, this is a very, very recent preprint that came out 2 days ago, conducted in Houston, Texas. They sampled 5,085 strains, and they found that almost every single one of them had the G614 variant. This is their sampling, and in early March, this is the G variant, and look at how it spiked starting June in these last 2 months. And this is a heat map between, this is March to May, and this is May to July. Look at how wide the distribution of the virus has become in Houston, Texas, and in what is known now as the second wave. The second wave is actually the G variant taking hold. And daily new cases, look, January, February, March, nothing, under 100,000 daily cases. But starting with the G variant taking hold, it's like exponential, and it's stayed up there. Now, this D61G mutation, to me, is very deadly because even though it doesn't kill you immediately, the D61G mutation mimics the open status in spike protein. Remember I said it has to be in an open or up state to bind receptors. This mutation mimics that open status. This is the D61G in the open position. This is the mutation. This is the normal virus in the open position. And when you superimpose this image over this image, they overlap completely. And what this means is that the mutation likely induced conformational modifications that changed antigenicity, possibly by mimicking the open and up state in the VAR-BD. Now, when it is in the open or the up state, it exposes the cleavage domains to proteases such as furin, TMPRSS, and cathepsin. And this is where furin comes in. Furin cleavage is important because it is widely expressed in human proteins. Take a look at how human protein expression is distributed in human tissues. That's a lot. And this is a measurement of the 26 of the 29 SARS-CoV proteins that have been found to interact with 332 human proteins. And what you're seeing is the top 16 of those 26 SARS-CoV protein abundance in human tissues. It's been found in lung, testes, fallopian tubes, adrenal glands, thyroid, tonsils, and the list goes on. Basically, it's everywhere. And these proteins that interact with the SARS-CoV virus are involved in multiple biological processes, including protein trafficking, translation, transcription, and even regulation of ubiquitination. This is a map showing these 26 SARS-CoV-2 proteins interacting and how they interact with 322 human proteins. And this is why you see multi-organ damage by the SARS-CoV-2 virus. And today, I'm going to focus on damage in the heart. Now, earlier I said the virus is detected in different cardiac tissues. And an autopsy of a child with MIS-C who died of cardiac failure, they found SARS-CoV-2 in different cell types of cardiac tissues. And not just in that autopsy. In another report on 39 autopsy cases, 61.5% of them had viral particles in the heart. And the viral load was quite large, with more than 1,000 copies per microgram of RNA in 67% of the patients. And there was also evidence of active viral replication. So how exactly does SARS-CoV-2 damage the heart? A group of scientists derived cardiomyocytes from stem cells, and then they infected them with SARS-CoV-2. And they found that these infected cardiomyocytes look very, very much like autopsy samples from COVID-19 patients, in that they showed a lack of nuclear DNA, and they also showed unique patterns of sarcomere disruption and fragmentation. Sarcomeres are long protein filament subunits in muscle fibers, and they are present in cardiomyocytes. Cardiomyocytes are muscle cells in the heart, about 25 micrometers in diameter and 100 micrometers, or 0.1 of a millimeter, in length. And cardiomyocytes contain myofibrils, and these are myofibrils. And in the myofibrils are small repeating subunits called sarcomeres. You can think of sarcomeres as the basic contractile units of cardiomyocytes that are found between two Z, what they call Z lines. And these sarcomeres in the Z lines are made up of myosin and actin filaments. So what do sarcomeres look like after they've been infected by SARS-CoV-2? Now, on the left, you see long, healthy fibers and heart muscle cells with the ability to contract. Healthy sarcomeres, healthy microfibers. And in the right are fragmented sarcomeres, or heart muscle cells, that are infected by SARS-CoV-2, like little bits, all chopped up and diced up. Long, stringy, healthy fibers and diced up fragmented sarcomeres. This is what SARS-CoV-2 does. And also, remember the nucleus? This is a healthy cardiomyocyte with a big round nucleus. And this is an infected cardiomyocyte with a completely enucleated nucleus that's empty and shriveled and displaced. So how do the SARS-CoV-2 virus get to these sarcomeres? Now, cardiomyocytes, they do express ACE2, which is a known receptor for SARS-CoV-2. And the expression of ACE2 is actually extremely low in other cardiac cell types in the myocardium. And the scientists that did the study on the stem cell-induced cardiomyocyte found that the infection and propagation was only found mainly in cardiomyocytes, but not cardiac fibroblasts and endothelial cells. But in the autopsy cases, SARS-CoV-2 viral particles and RNA was detected in the myocardium. And so that means something else could be the receptor for these cells, because cardiomyocytes only express ACE2. But if these other cells in the myocardium are infected and they don't express ACE2, what could be another receptor for these cells? Now, ACE2 is now known and being discussed in a lot of papers that their expression actually starts to decrease, starting from the nose to the lungs, and the infectivity actually decreases. ACE2 expression decreases as you get to the lungs. In fact, they find very, very little ACE2 expression in lungs. And the ACE2 protein expression in heart muscle is actually 12-fold less than in small intestines. ACE2 expression is the highest in small intestines. Heart is here. The level is 10.5, in small intestines is 122. So ACE2 the expression is really low in the heart. And remember, they found SARS-CoV-2 in different cell types, not just cardiomyocytes in the cardiac tissues. Now, in May, there was a paper that discussed a role for something called Integrins in host cell entry by SARS-CoV-2. They said Integrin may be an alternate receptor, and they actually presented a very convincing case because the spike protein of SARS-CoV-2 has a new RGD motif that can bind integrins. And this motif is actually absent from other coronaviruses. Now, this is the receptor binding domain. right here, starting from protein number 319 to 514. This is where receptors get bound to the virus. And this RGD motif is right around protein number 386, and is very close, almost next to the area that binds ACE2, 437 to 508. Now you notice, these are all different kinds of coronaviruses. They all express ACE2 binding motifs. But only the SARS-CoV-2 has an RGD tripeptide sequence that can also bind integrins. So what is beta-1 integrin? Now, first of all, it is encoded by the ITGB1 gene, and they are ubiquitously expressed cell surface receptor adhesion molecules that are found in blood, immune cells, and every major organ of the human body. Take a look at the protein expression of integrins everywhere. And integrins are also found in all subcellular compartments, including mitochondria. Subcellular compartments, including plasma membrane, extracellular, endosome, cytoskeleton, nucleus, endoplasmic reticulum, mitochondrion, cytosol, lysosome, Golgi apparatus, and peroxisome. And now, does it start to make sense that 26 of the 29 SARS-CoV-2 proteins can interact with 332 human proteins? And again, protein expression of ACE2 is very, very low in human tissues. So it doesn't make sense that ACE2 is the major receptor. There must be another receptor that brings the virus to all the other human tissues. Look at all the human tissues that have been found with SARS-CoV-2 protein. And what is interesting is beta-1 integrins, they are absolutely necessary for the proper assembly of sarcomeres. Remember sarcomeres and how the virus chops them up? Now, beta-1 integrins, they bind to both ACE and ACE2. And that binding allows them to act as cell adhesion substrates through the common RGD tripeptide sequence. And that's how ACE2, ACE, and integrins work in the cells to function as adhesion molecules. Now, this RGD binding motif, it recognizes integrins to promote cell adhesion. And it is also known that this motif facilitates viral internalization through the activation of transduction pathways. So this is not something new. This is something that has always been known about integrins and what they do in viral transmissions. Now, increased ACE2 expression enhances adhesion processes, and ACE2, when they bind to integrins, they can regulate cardiac remodeling signaling pathways that can affect cell survival and proliferation. So you can imagine when the virus actually binds integrins and ACE2, they take them out of circulation. So that becomes a major disruption in cell survival and proliferation. And this RGD motif is present in both the ACE2 receptor and the spike protein of SARS-CoV-2 that's close to the receptor binding motif that binds ACE2, which I just showed you earlier. This RGD motif here is right next to the area that binds ACE2. So you can have the virus binding integrins, and then it'll conveniently bind up ACE2 also if it is around. Now, beta integrins is really very important for the maintenance of healthy heart muscle fibers, because skeletal muscle development and functions are actually dependent on beta-1 integrins. All muscle fibers express the integrin beta-1D isoform, and in heart muscles, the integrin beta-1D is the only isoform expressed in adult cardiomyocytes. What most people don't know is integrin beta-1 delta is uniquely downregulated in a hereditary disease called arrhythmogenic right ventricular cardiomyopathy, ARVC. Integrin beta-1D deficiency is the mechanism that causes arrhythmias in ARVC. And integrin beta-1D deficiency actually leads to critical structural changes that affect the connection of protein structures in sarcomeres. Now, I want you to take a look at the striking similarity in heart muscle fibers from patients with beta-1D deficiency and the muscle fibers that were infected by SARS-CoV-2. This is SARS-CoV-2 infection of sarcomeres, and the fragmentation, little bits all chopped up. And these are patients, ARVC patients, who have inherited a genetic deficiency in integrin beta-1 delta. And this is a healthy donor, a comparison. Look, little bits of sarcomeres all chopped up. Take a look again. This is a slightly blown up, so the bits looks larger. But if you look closely, all little bits are chopped up. And remember, 39% of cardiac disease in hospitalized COVID-19 patients show right ventricular dilatation, right ventricular dysfunction. Major symptom, right ventricular functions are impaired. Impaired right ventricular contractile function. So integrins and sarcomeres are deeply tied to right ventricular dysfunction. So how does integrin affect sarcomeres? A decrease in integrin-mediated adhesion in adult muscles led to a progressive loss of muscle function due to a failure to maintain normal sarcomeric cytoarchitecture. And reduction in integrin-mediated adhesion resulted in detachment of actin filaments from the Z lines, and separations of the Z lines from the membrane eventually led to disintegration of the Z lines. And when you have disintegration, you get fragmentation. All the Z lines are disintegrated. So your sarcomeres, basically, there's nothing to hold up the sarcomeres, so they end up with little bits and pieces. And you can find many references in literature where the differentiation and integrity of cardiac muscles are impaired in the absence of beta-1 integrins. And endothelial beta integrins regulate the sprouting and the network formulation during vascular development. Now, endothelial beta-1 integrins. Remember endothelial cells? And that's how SARS-CoV-2 infects endothelial cells in the in the heart muscles using beta-1 integrins. To me, heart damage after the recovery from COVID-19 infection is actually a major concern going forward. And people are starting to pay attention. You see, doctors are trying to understand why and how COVID-19 can damage the heart. Now, this is a preprint that looked at 139 healthcare workers who were examined 10 weeks after their SARS-CoV-2 infection. 75% had abnormal cardiac MRI, 50% had abnormal electrocardiograms, and 40% fulfilled the criteria for either pericarditis or myocarditis, and 26% of them had myocarditis. And pericarditis and myocarditis were present in even subjects that were asymptomatic. And the implications of viral myocarditis in COVID-19 really requires urgent clarification. People do talk about COVID-19 and myocarditis, but they really have not come to any type of consensus or conclusion. But they do recognize that myocarditis is a direct viral involvement of SARS-CoV-2. But then they believe that SARS-CoV-2 used an ACE2 to bind to spike protein. And if you only look at ACE2, you're really missing the big picture on integrins. Because integrin receptors, they mediate direct viral entry to cause ventricular dysfunctions and myocardial injuries. Myocarditis is an inflammatory disease of heart muscles that has been reported in COVID-19 patients. But the thing is, it's not only in SARS-CoV-2 infections, because more than 20 types of viruses have been associated with myocarditis. The pathophysiology of myocarditis is a combination of direct viral infection and also cardiac damage from host immune responses. And viral myocarditis can cause ventricular dysfunction and sudden cardiac death in children and adults under the age of 40. And I'm sure you have heard many reports of people who seem perfectly fine, they got COVID, and the next thing you know, they had a heart attack and died. I've heard these stories many times. This is how viral myocarditis works. It can cause ventricular dysfunction and cause sudden cardiac failure and cause death in children and adults under the age of 40. That's a very specific feature of viral myocarditis. And integrins are known to play important roles in viral infection by facilitating viral entry. Now, in people, or actually in the case of the child who died from cardiac failure after admission to hospital with MIS-C symptoms, one of the symptoms she manifested was actually hypotension with blood pressure of 80/36 and tachycardia 134 beats per minute. And this is actually a report all the way back in mid-April, when they first found children with MISC in the UK. And that was the time when they compared it to Kawasaki disease. And all of these children had hypotension that required vasopressors such as noradrenaline, dopamine, and noradrenaline as pharmacological interventions. Look at 80/40, 81/37, 90/30, 77/41, 85/43, 77/46, 80/48, and 82/41. Now, normal children between the ages of 6 to 12, their systolic should be around 90 to 100, and their diastolic should be 70 to 80. So with the diastolic hovering around 35 to 40 something is really, really low. Now, adult COVID-19 patients have also presented with hypotension, but clinicians actually can't figure out why, because when you have systemic inflammatory responses, you usually have hypertension and not hypotension. Well, guess what? Right ventricular myocardial dysfunction can cause hypotension, severe hypotension. Right ventricular failure manifests as hypotension. So what is right ventricular failure? It occurs when the right ventricle cannot support adequate blood flow through the pulmonary circulation to maintain adequate left ventricular filling. Now, when that happens, and it can occur suddenly in a previously healthy heart due to either massive pulmonary embolism or myocardial infarction of the right ventricle. And hypotension is a hallmark of right ventricular failure, as the tachycardia and increased stroke work creates substantial RV free wall tension that increases oxygen demand. This eventually would decrease cardiac output and results in reduction of blood pressure. And right ventricular failure can be a direct result of myocardial disease or myocarditis. And beta-1 integrins is uniquely deficient in arrhythmogenic right ventricular cardiomyopathy. Beta-1 delta integrin is downregulated in hereditary arrhythmogenic right ventricular cardiomyopathy. And this deficiency is the mechanism that causes arrhythmias. And this deficiency leads to structural changes that damages the protein structures in sarcomeres. And I've shown you before that they almost look exactly alike, the sarcomeres from ARVC patients and those that were infected by the SARS-CoV-2 virus. So this brings us to the conclusion of the first part of our presentation today. And we'll take a break for some questions if anybody has any questions on the materials I presented so far. And then the second part, I'll focus on how ancient molecules can help us live better with SARS-CoV-2, protecting our hearts.
I have a couple of questions that have come up in the chat while you were doing your first presentation. And one of them has to do with the question of potentially strengthening sarcomeres against this kind of destabilization. And I'd like to know if you've run across anything that indicates that there's a collagen dependence on these fine muscle fiber structures.
Now, you're talking about two different things. You're talking about the substrates, and you're talking about what actually glues them together. So if you're talking about collagen, yes, if you lack collagen, you will basically have very poor muscular structure everywhere. It doesn't pertain to only sarcomeres. What is important with the sarcomeres is how they are being held together by beta-1 integrins. And so in part 2, I will actually talk about how sarcomeres can be strengthened. But of course, vitamin C, collagen, and anything that strengthens and builds fibers and muscles are important.
How about any contributory role of local glutathione concentrations or the glutathione redox couple?
Okay. Now you're talking about oxidative stress and how it damages cytoarchitecture. I think specifically for sarcomeres. Now, the redox environment always affects the health of all the cells. So that is a given. And if you are deficient, and if you have high oxidative stress, that will contribute to lower performance. But with everything being equal, I don't think particularly increasing glutathione, if you are missing the important glue function of the beta-1 integrin, you would be able to turn around the situation. Does that make sense?
Yeah, but I'm not sure if it really goes to the heart of the question that I'm asking.
Oh, okay.
About whether or not there's that there's a way in which environmental changes in adjacent tissues can have, in a sense, catastrophic processes on other ones where the researchers aren't necessarily looking.
Bill,
who's talking?
property. If I give you the address, can you determine that for me?
Janet, please mute yourself.
733 Harvard Avenue.
Wow, sorry about that. That when researchers are looking at particular dimension of a pathology, they may not see something that's going on.
Right. Okay, I see what you're talking about. Yes. Now, if you go into EMF and its effects, yes, it definitely affects all the cells. And how do I explain that? So is the question, does glutathione protect a cell in a high EMF environment? Is that the question?
Not at all.
Okay, I still don't get the question.
It was more about, is there some kind of oxidative stress that is a required condition for the disassembly of the myocytes and the damage that's taking place that is not actually being measured when the researchers are going in and looking at the situation, so they don't understand the interconnectedness of different phenomena.
Right, okay. In my current research on sarcomeres, the disintegration is really not directly caused by oxidative stress. It is caused by a lack of deficiency in the beta-1 integrins. But oxidative stress can impede any and all metabolic functions. So when you talk about EMF and oxidative stress, it is a factor that needs to be considered in every equation, because a person will heal better in a low EMF environment. That there is no question about that. Or if a person has less oxidative stress, they will recover faster than a person with poor redox.
So is the Integrin system in any way dependent upon the redox environment in which it's operating? Or the presence of vitamin C is, let's say, a cofactor or involved in some kind of Fenton chemistry?
Yes, yes. The next part, we'll talk about what actually can increase integrins, beta-1 integrins. And I'm actually quite excited. And that is also actually why vitamin C and melatonin has been found to be so helpful for almost everyone who's been infected with COVID-19 and took these two dynamic duals, as I call them. And it just worked for so many people. I mean, I have gained a lot of followers, and at first I didn't know why. And then when people start telling me how using ascorbic acid and melatonin has helped them not only reverse disease of the infection, but a lot of them have started to actually reverse their basic underlying health challenges. And that was a surprise for many people. And a lot of them are older from the older generation. So very, very exciting.
So let's go on to part two then. I don't think we've answered all the questions at this point. And if somebody wants to raise their hand and ask a question, please do.
There is one more question. Levy had a question that he has an MTHFR gene mutation that leads to excessive clotting. And he was wondering if Doris could comment on this in reducing the complications of COVID. And the question is answered. You can do that either now or later.
Probably later.
Okay.
Yeah. Okay. So let's go to screen share.
And we're back.
Yes. Okay. Ancient molecules for a new virus. Living with SARS-CoV-2, the heart of our future. Now, SARS-CoV-2, I'm really sorry to say, is really here to stay. And sooner or later, pretty much everyone will be infected by it. Some people may not show any symptom, and other people may have a more difficult time. But now, even though I said a new virus, because this virus is new in that even though it shared a 79% similarity with SARS-CoV-1, there are genetic variations that are different and never seen before. So even though this is a new virus as a kingdom is actually very, very old. In fact, some scientists think that they share the same ancient ancestors as all the other living organisms in the three other kingdoms. This is over 3.8 billion years ago with the origin of life that spawned viruses, archaea, bacteria, and eukarya, and we are eukaryotes. So why am I focused on ancient molecules? If viruses have been around 3.8 billion years ago, You would think that since they're so nasty and sneaky, they probably wipe out all the other kingdoms. No, actually, every kingdom are still alive and well. Bacteria and eukarya, we are all here today. And we existed and evolved with viruses. So there must be something that protected us from the viruses and helped us to adapt to these viruses as they infect us. So the solution to me is actually not new drugs that can do this or that, but something that's been around for over 3 billion years that evolved together with all the three kingdoms, together with viruses, protecting the eukaryotes, and that is our ancestors and us. Melatonin is an ancient molecule, over 3.5 billion years old. It's 3.5 billion years old in prokaryotes, in the plant kingdom, and the animal kingdom. Now, most people think of melatonin as a sleep hormone. And in the beginning, it was just an antioxidant that protected life. It did not gain all the other functions that we know it is capable of doing, which is regulating immunity, circadian rhythm, sleep, and anti-cancer properties, until, let's say, about 600 million years ago. There was like over 2 billion years of evolution here that is outside of what we know of it today. Now, ascorbic acid is also a molecule that's over 3 billion years old, but it is not quite the same as melatonin, because melatonin is an antioxidant. And for people who think of ascorbic acid as an antioxidant, they are missing quite a lot on what this molecule can do. It is a redox molecule in ancient eukaryotic organisms. And this is a diagram of the evolution of ascorbic acid from the very, very first eukaryotic ancestor, over 3 billion years ago. Now, humans and primates have actually lost the ability to synthesize ascorbic acid, but they do produce melatonin. Now, melatonin production in humans goes through stages. It is actually the highest in children under 10 years old. And as you grow older, starting around 40, the level starts to drop. And by the time you get to 60, you basically produce a negligible amount of melatonin. Now, remember before I said SARS-CoV-2 affects multi-organ damages. And acute kidney injury is something that's very, very, very common in COVID-19. And people started to document acute kidney injury all the way back in May already. May. And acute kidney injury documentation continues, July. In September, it is getting worse. The incident, risk factors, and prognosis of acute kidney injury in COVID-19. And guess what? Melatonin actually improves renal functions in chronic kidney disease patients. Now, chronic kidney disease patients that have the worst renal function of GFR under 30 millimeter per minute had the lowest melatonin concentrations in their blood. And the CKD patients with the best renal functions have the highest circulating melatonin levels during peak mid-pineal production hours, which is 3 a.m. This is the difference between patients with the best renal functions and the ones with the poorest renal functions. And systemic inflammation is a hallmark of acute kidney injury. So how does melatonin protect kidneys? If systemic inflammation affects kidney injury, melatonin must be able to do something with the immune system. Well, melatonin actually enhances pro-inflammatory immune responses. Now, in COVID-19 patients, elderly individuals, especially men, they showed weaker immune responses that were associated with worse clinical outcomes. And older individuals had a 30-fold reduction in the expression of a chemokine called CXCL11. This chemokine is induced by Th1 and interferon gamma. And this chemokine is responsible for directing the trafficking of T cells and natural killer cells. So if you don't have enough of this chemokine, your T cells and natural killer cells basically don't know where to go or what to do. But melatonin has been shown to be able to enhance the release of interferon gamma that's produced by Th1 immune cells. And with the increase of this interferon gamma, you would end up with having more CXCL11. On top of that, melatonin also stimulates the production of natural killer cells and lymphocytes by promoting their differentiation from progenitor cells. Now, at this point, I'm sure a lot of you are wondering, well, if melatonin enhances pro-inflammatory immune responses, what happens when they go out of control and you end up with cytokine storms, which is uncontrolled inflammatory responses? Now, this is where the ancient aspect comes in. Now, if melatonin can increase the pro-inflammatory responses, it also probably would be able to balance that by inhibiting the immune responses so that you don't end up killing the organism. And this is what I mean by something ancient that has evolved with the virus so that it knows what to do under what circumstance. And a lot of people actually call melatonin a smart molecule because they find it to do different things under different circumstances, or they find it to do opposite things under different circumstances. Now, melatonin can actually inhibit cytokine storms through the NLRP3 inflammasomes. The activation of NLRP3 inflammasomes can initiate cytokine storms in COVID-19 patients. And monocyte-derived M1 macrophages, they are the ones that can activate these NLRP3 inflammasomes and secretes pro-inflammatory cytokines, including interleukin-1 beta. Now, melatonin suppresses NLRP3 inflammasomes by upregulating the expression of NRF2 and also by regulating macrophage phenotypes. Now, there are basically two pools of melatonin in vertebrates. There is one pole that is produced in the pineal gland that is regulated by light-dark circadian signals. And there is a non-circadian pool that is produced in mitochondria of all cells. Every single cell that has mitochondria will produce melatonin. Melatonin that is produced in mitochondria is actually localized and is not released into circulation, unlike the pineal melatonin, which can be released into the cerebrospinal fluid and blood. Now, all macrophages, they contain mitochondria. And the pro-inflammatory M1 type macrophage mitochondria, they do not produce melatonin, whereas the anti-inflammatory M2 macrophages, their mitochondria produce melatonin. Now, how is that? Now, we first have to look at what melatonin does in mitochondria. Now, mitochondria produce melatonin, and this is the pathway, and this is exogenous melatonin, which is melatonin you take in supplements, which can go through transporters into the cytosol. Now, inside the mitochondria, melatonin scavenges reactive oxygen species and increases antioxidant activities by upregulating SIRT3 and superoxide dismutase, SOD2. And you end up with less oxidative stress and redox homeostasis. Melatonin also upregulates oxidative phosphorylation and increases acetyl-CoA. Now, before I said melatonin can stop cytokine storms by converting macrophage phenotypes. What are macrophage phenotypes? Classically activated M1 macrophage, they produce pro-inflammatory cytokines, reactive oxygen species, and nitrogen species to create an environment that is high in oxidative stress that can kill pathogens. However, if you cannot convert the M1 back into the anti-inflammatory M2 phenotype, you end up with uncontrolled inflammation and cytokine storms that are often reported in severe COVID-19 patients. But melatonin can convert the polarization of macrophages from the M1 pro-inflammatory to the M2 anti-inflammatory phenotype. So how do cytokine storms develop? Monocytes from progenitor cells in the liver are transformed into basically two types of macrophages during active infection. The pro-inflammatory M1 uses aerobic glycolysis to produce ATP, and they also produce large quantities of cytokines. Look, this is all the cytokines and chemokines contributing to the development of cytokine storms. Whereas the anti-inflammatory M2 macrophage use mitochondrial oxidative phosphorylation, OXPHOS, to produce ATP and only a small amount of cytokines. And melatonin, whether it's exogenous, you take in supplements, or endogenous, can convert M1 into M2 and stops the production of cytokine storms. And the key where that switch takes place is actually inside the mitochondria. Under non-inflammatory conditions, macrophages basically employ OXPHOS to support their normal functions. Pyruvate is a substrate that is converted from glucose in the cytosol and then transported into mitochondria. And there is an enzyme called PDC, pyruvate dehydrogenase complex, which then converts this pyruvate into acetyl-CoA. Now, acetyl-CoA is very important because it supports oxidation via the citric acid cycle, also known as the Krebs or the TCA cycle. And what is even more important is acetyl-CoA controls melatonin synthesis in mitochondria because it is a required substrate for the rate-limiting enzyme AANAT. So now I'm going to show you how exogenous melatonin can increase endogenous production of melatonin in mitochondria via the PDK and the acetyl-CoA pathway. Many of my readers and friends have said to me, "Hey Doris, does taking melatonin stop endogenous production?" And I tell them, no, I have never seen any study that says taking exogenous melatonin can stop endogenous production in the pineal glands. But what I didn't tell them is, if you take exogenous melatonin, you can actually increase endogenous production of melatonin in the mitochondria. This, let me explain, this is the substrates for endogenous production. This is the pathway, and this is ANAT, the rate-limiting enzyme that ends up with melatonin. And this is your exogenous melatonin that you take, that is being taken up into the mitochondria. In M1 macrophage, the PDK enzyme, the pyruvate dehydrogenase kinase, is actually activated. PDK is activated in mitochondria of M1 macrophage. And when PDK is activated, what does it do? It stops PDC. Stops it dead in the track. Without PDC to convert the pyruvate, you won't have acetyl-CoA. And without acetyl-CoA, you basically have downregulated oxidase and lowered ATP. And that is how M1 macrophages end up using, or they prefer to use, aerobic glycolysis. And that's how they use aerobic glycolysis, because they will activate the PDK to stop the PDC and stopping everything in the track. And stopping acetyl-CoA, if you remember, will stop melatonin production because it is a required substrate for ANAT. And that is why mitochondria in M1 does not produce melatonin. However, if you take exogenous melatonin, it will stop PDK, reactivate PDC, and everything will go back into that state where you have oxidase and endogenous production of melatonin and the mitochondria phenotype that supports M2. And that is how melatonin and exogenous melatonin converts M1 into the anti-inflammatory M2 phenotype. Now, what I have shown you just now is really a very oversimplified version of what happens. The conversion of M1 into M2 by melatonin in mitochondria is actually a very complicated process. And I suggest you read this paper if you want to find out exactly how melatonin does it. And the major pathways affected by melatonin during this conversion includes PDH deactivation, HIF1A suppression, glutaminolysis, elevation of alpha ketoglutarate, enhancement of complex 1 and 4. Now, this is very, very important because most people don't know that melatonin can increase the expression and the activities of complex 1 and 4 in mitochondria. And melatonin can also increase fatty acid oxidation. And melatonin is now, 2020 paper, recognized as being cardioprotective. Even in COVID-19, melatonin is now recognized as something that can protect against myocardial injury. And this is a wonderful paper by yours truly that talks about how melatonin can protect humans from myocardial injury. Now, this paper actually focuses on the blood clotting aspect, rather than what we're talking today. So, Steve, before you said, how do we affect beta integrins? Is there something that can help increase beta integrins? Well, this is your answer, melatonin. Melatonin actually increases the expression of beta-1 integrins. After 24 hours incubation, there is a twofold increase, double beta-1 integrins, with melatonin and without melatonin, no change. After 24 hours, boom, you have twice the amount of beta-1 integrins. Now, when SARS-CoV-2 infects a cell and binds to beta-1 integrins, it basically takes it out of circulation. So with less and less beta integrins to act as adhesion molecules, basically like, you can think of it like glue, your sarcomeres are going to disintegrate. The fibers are going to get, there's nothing to hold them together. You can think of it that way. And so if you take melatonin, you actually end up with more beta integrins that will help you through the period when you have a lot of binding to the virus. So we know that beta-1 integrins modulate sarcomere assembly, and it actually can facilitate muscle regeneration and improve muscle function. So if beta-1 integrins can do that, and if melatonin increases beta-1 integrins, does that mean that melatonin can also help muscle regeneration and improve muscle functions? Oh yes, 2020 paper. Melatonin has been found to reduce skeletal muscle frailty and increase physical performance. Now, this is actually very true because I've had reports from people who use melatonin and they say, hey, Doris, this is amazing. I haven't exercised, but I can do more, lift more, run better, run longer. My muscles are just incredible. I can lift so much more without getting tired. Well, basically, melatonin is rejuvenating all your muscles. And sarcomeres are present in all muscles, and so is beta-1 integrins. And if it can do that for your arm muscles, it will do that for your heart muscles. And look, melatonin preserves normal muscular activity structure and the amount of total muscle fibers in old mice. Now, old mice and old humans, they have what is called sarcopenia, which is a loss of muscle fibers and integrity and total muscle fibers. But melatonin can prevent that from happening. Now, this is a great study that showed melatonin can really protect sarcomeres in cardiotoxicity situations where, now this is not a paper on viral infection, but it is cardiotoxicity by an anti-cancer agent known as doxorubicin. Now, doxorubicin has known side effects, including the destruction of myofibrils, disorganization of sarcomeres, and mitochondria degeneration. But what they found is, when you add melatonin to doxorubicin, the treatment reduced all the structural changes caused by doxorubicin in myofibrils and sarcomeres. So now we go on to vitamin C. Viral myocarditis is inflammation of the heart muscles caused by viral infections. And in children, when you add intravenous vitamin C to conventional treatment therapies for viral myocarditis, they end up with significantly better results. Why is that? That we go back to the lab results of that poor 11-year-old COVID-19 patient who died from cardiac failure from MIS-C symptoms. She had very high levels of white blood cell count, extremely low lymphocytes, elevated D-dimer and troponin, and extremely elevated ferritin levels. The normal range is 20 to 200. She had 1,501. Now remember, within 24 hours of admission, she died, and these are the lab results taken at several intervals during these 24 hours. Ferritin. Why is ferritin elevated in her? What does ferritin do? Ferritin sequesters oxidized ferric iron from toxic free heme. When red blood cell normally auto-oxidizes and produce a certain amount of free heme. But when you have systemic inflammation, if you have viruses attacking your blood or high oxidative stress situations, your red blood cell may be compromised and you will have free heme where the iron, instead of the stable ferrous form, becomes the oxidized, unstable ferric form. Now, free heme is toxic. And when that happens, these free heme, they will be bound by haptoglobin and engulfed by macrophages. These macrophages will gobble these up and metabolize them so that the ferric iron can be incorporated into ferritin. And you can think of ferritin as the primary site of iron storage in a cell. And if you have severely elevated ferritin, that means you have a lot of toxic free heme floating around that contains oxidized, unstable ferric iron. This is what happens in COVID-19. Now, high mortality rates are often associated with hyperferritinemia, which is a marker also for significant macrophage activation. And this often leads to multiple organ dysfunction. Elevated ferritin can accurately predict fatality levels in COVID-19 patients, where non-survivors had elevated mean ferritin levels of 1,297.6 nanograms per milliliter, which is more than double the 614 nanograms per milliliters in survivors So non-survivors had double the amount of ferritin. And take a look again at the lab results. Non-survivors, 1297. What does she have? 1501. Now, free heme, it leads to the perfect cytokine storm because excess free heme leads to excess ferritin and macrophages. And before, I told you macrophages can cause cytokine storms unless you have enough melatonin. So how does and where does ascorbic acid come in this free heme ferric iron equation? Red blood cells, erythrocytes, they use ascorbic acid as electron donor to maintain heme in the ferrous stable form. Now remember, in the very, very beginning, I said ascorbic acid is an ancient molecule and it is not an antioxidant. It is a redox molecule because it can donate electrons and then accept electrons. And that means it can be regenerated. So this is a heme. And this is the reduced form of ascorbate. And it donates an electron to keep the heme in the stable form. But what happens to this ascorbate that lost one electron? It becomes oxidized. And the form is now called AFR, or monodehydroascorbate. This is where Cytochrome B5 Reductase 3, CYB5R3, comes in. CYB5R3 is a redox enzyme that specifically targets ascorbic acid and nothing else. CYB5R3 has a soluble form that is found exclusively in erythrocytes. And that's how erythrocytes regenerate ascorbic acid, to keep the heme in the reduced form, to keep it from being free and toxic. There are basically two forms of cytochrome B5 reductase 3. The membrane-bound form is ubiquitously expressed in all mammalian cells. Every single cell has CYB5R3. And anchored to the outer mitochondrial membrane, the OMM, or put it this way, the membrane-bound form is anchored to the outer mitochondrial membrane, the endoplasmic reticulum, and also the plasmatic membrane. And the function of the CYB5R3 is to facilitate the rapid conversion of oxidized ascorbate back into the reduced ascorbic form using cytosolic NADH as electron donor. If you have decreased expression, or if the enzyme is inhibited, you will end up with mitochondrial dysfunction, reduction of ATP synthesis, and a lowering of the NAD plus NADH ratio, and as well as increased sensitivity to oxidative stress. Now, a lot of people know that having more NAD plus is good. But NAD plus and NADH is always in a ratio that's balanced. If you have more NADH than you have, well, less NAD plus. And that sometimes can put stress on cellular metabolism. And what CYB5R3 does is it maintains this ratio to keep the NAD plus high by using NADH to regenerate ascorbic acid. And that's why when you take ascorbic acid, you actually end up with more NAD plus. And usually people say when they take ascorbic acid, they are more energetic. And you don't actually end up have to take those expensive NAD plus supplements because ascorbic acid will naturally increase your NAD plus ratio. It uses NADH to regenerate ascorbic and you end up with NAD plus. This is the reduced form and this is the oxidized form. And this is the methemoglobin, which is the free form of heme that has the oxidized iron. And if you donate a electron, you will reduce the methemoglobin into regular hemoglobin. But then this oxidized ascorbic acid needs to be regenerated by CYB5R3 using NADH, and NADH becomes NAD. And if you stop this function, you will end up with mitochondrial dysfunction, decrease of the NAD-NADH ratio, and oxidative stress. And these are the locations of Cytochrome B5 Reductase 3. The soluble form is inside, and the membrane-bound form is on the plasma membranes, the outer mitochondrial membrane, and the endoplasmic reticulum. And would you know that CYB5R3 is critical for maintaining redox balance in cardiomyocytes? Adult cardiomyocytes that are deficient in CYB5R3 being experimented in mouse models, they show increased oxidative stress, decreased cytochrome coenzyme Q levels, and increased heme iron oxidation. And CYB5R3 knockout mice, they suffer sudden cardiac death associated with calcium mishandling, increased oxidative stress, decreased ATP production, and loss of hemoprotein redox balance. Now, how does CYB5R3 protect cardiomyocyte? Basically by two functions. It maintains the heme, the stability of the heme, and it protects the mitochondrial functions so that cardiomyocytes have enough energy to perform optimal functions. Now, CYB5R3 maintains heme iron in the stable reduced ferrous state. And that is why CYB5R3 is also sometimes called the methemoglobin reductase, because it lowers the amount of methemoglobin formed during erythrocyte auto-oxidation events. And mice bred without CYB5R3 cannot reduce heme in their erythrocytes. What's more, they also had smaller mitochondria that produce less ATP due to reduction in oxphos from the impaired expression and activity in complex 1, 2, and 4. Now, lowered oxphos is really problematic because it can reduce oxygen pressure in cardiomyocytes by creating a pseudo-hypoxic state. And CYB5R3 knockout mice had more than 50% lethality with increased left and right ventricular stroke volumes, decreased ejection fractions, myocardial inflammation, and note, myofibrodystrophy. And I'm sorry to say that humans with mutated with CYB5R3 genes are associated with accelerated time to first acute cardiac event and accelerated time to death. Now, we've seen this before, and what I want to emphasize is how macrophages, again, engulf these free heme. Macrophages also recycle high quantities of ascorbic acid. They accumulate ascorbic acid at rates up to 100-fold using their special transporters called SVCT2 and also GLUT1 transporters. Now, why do macrophages want so much ascorbic acid? Macrophages actually control their own levels in the right condition and environment. They use ascorbic acid to generate oxidative bursts to kill monocytes. And if you remember, monocytes are the parents of macrophages. So if you kill monocytes, you're basically killing your next generation of macrophages. Macrophages use ascorbic acid to produce nitric oxide. And this nitric oxide is what they use to generate the oxidative bursts. This lethal oxidative burst can kill not only surrounding pathogens, but it is part of a negative feedback loop where that oxidative burst also kills the monocytes, which are the parents of the macrophages. And nitric oxide deficiency in macrophages from insufficient ascorbic acid is probably one of the main reasons how cytokine storms can be exacerbated in COVID-19. And in severe ARDS cases in COVID-19, you usually find high levels of monocyte-derived macrophages. And if you go back to that lab report of that 11-year-old MISC patient, there was extremely high levels of white blood cells. And how does oral ascorbic acid increase nitric oxide? It's a compound called BH4. It is the rate-limiting enzyme in nitric oxide production, but BH4 can oxidize very quickly. And it turns into the oxidized states that will not only not produce nitric oxide, but also destroy nitric oxide. So BH4 requires the presence of ascorbic acid to generate more nitric oxide, because ascorbic acid acts as a redox molecule to donate an electron to keep BH4 in the reduced state. And in nitric oxide synthesis, ascorbic acid is almost used exclusively in BH4 recycling. And there have been studies where they found other antioxidants like thiols, they are actually ineffective in regenerating BH4. So this comes to a summary of what these ancient molecules can do for healthy hearts. SARS-CoV-2 infections can cause subclinical myocardial damage long after recovery. And this is my main concern. It is no longer about death rates. With the high infection rate and more people contracting SARS-CoV-2 virus, what do we really know about the subclinical myocardial damage that can affect someone months or even years down the road? Acute myocardial injuries can affect children and young adults without warning or symptoms. And if we use ancient molecules like melatonin, they can protect cardiac muscles by increasing the expression of integrins. And melatonin during infection also reduces cytokine storms by inhibiting NLRP3 inflammasomes. And what is more important is that melatonin can convert pro-inflammatory M1 macrophages back into the anti-inflammatory M2 phenotype by increasing oxphos in mitochondria. And even if you are not sick, increasing oxphos gives you more energy. Ascorbic acid can lower toxic heme production by providing electrons to support CYB5R3 functions and protect cardiomyocytes. And ascorbic acid can maintain macrophage balance by supporting the oxidative burst production via nitric oxide synthesis. And finally, to understand how SARS-CoV-2 damages red blood cells and why ascorbic acid and melatonin can attenuate vascular and endothelial dysfunctions, please follow these links and read all these articles. And melatonin can have really complex functions that help us defend against, help protect us and defend us against the SARS-CoV-2 virus. And this is the paper that describes these functions in detail. And that is it for today. And thank you very much for your time. And I hope you've learned something. And I hope all of you will take melatonin and ascorbic acid to protect your hearts, with or without SARS-CoV. Thank you.
You're welcome. Fantastic presentation. Can you talk a little bit about the practical aspects of supplementation that you have, any insights you have about taking melatonin?
Okay, melatonin. If you're not sick, you don't need a high level. And it also depends on your age. Remember, I showed you the chart where children have more and adults have less, and the older you are, the more you need. But however, even in adults, I've found that in working with people, one 70-year-old may need 500 micrograms, another 70-year-old may only need 100 micrograms. So you have to experiment. So how do you know? You take melatonin at night, after sundown, preferably 1 to 2 hours before you go to bed, but I would say preferably before 10 p.m. After you take it, if you wake up the next morning refreshed, energetic, you've taken the right dose. If you wake up groggy and takes you a while to get your bearings straight, you've probably taken a little bit over what you need. So these are some of the more obvious reports I've encountered. I myself can take a lot and it doesn't matter.
Any insights into why really high dose melatonin acts as a birth control agent?
Yes, because it suppresses estrogen.
Okay.
That's why during puberty, melatonin drops like a ton of bricks.
Okay, I'm looking for questions. We don't have any hands raised. We're getting all kinds of awesome thank yous, wonderful presentation, all that kind of stuff.
Now, I actually saw a question from Paul Fendler. He says, how do you know if you're taking too little melatonin? That is actually a very good question. Now, I would say the way you tell is actually not as immediate. If you don't feel anything different, then you're probably not taking enough. And I would say within 1 week, you should start seeing results. And some of the results are really pretty remarkable. Now, I have never been able to exercise because my health has always been not very good. So I'm the type, if you exercise, I get too much oxidative stress and I get sick. So before I used to get a headache if I lift a 1 gallon jug of water. And after I've been taking melatonin, I found that I can lift this is recent, this is just a few days ago, I lifted a 17-liter jug of water from the ground to the counter and poured it into a dispenser without feeling anything. And this is the same thing people tell me. They say, wow, I can lift a couch and, you know, I moved my furniture and I wasn't tired. I had no sores, no aches. And it's just, this has to be the melatonin because I didn't do anything different. And they want to know why. I said, well, wait for the presentation. You'll find out why, because melatonin actually protects and regenerates muscle fibers. Very simple. Integrins. And that's the same thing. That's going to help your heart. And please take melatonin for your heart.
Susie, go ahead with your question.
Yes, about a year ago, I had purchased something that I'd put on my head and it would shine a light in my eyes from 7 a.m. to 9 a.m. in that range for like 20 minutes. And that was to stop the melatonin production in the daytime, I think. And then I noticed at night I would like fall asleep. Is that a legit way to do it?
Yes. Well, you actually don't need to do that because any bright light in the morning will stop melatonin production. Now that is actually a very, very important aspect because this is where we're going to talk about the EMF bit. Now, we know that light at night disrupts melatonin production, and we've had light at night for hundreds of years. And in a recent article, I've described how the equivalent of 3 candles can reduce the production of melatonin by 10%. So imagine your LEDs, all your lights. I mean, it's like no melatonin. And what our bodies have started to do over these hundreds of years is actually to mutate. And if you do some Google searches, you will find that the melatonin receptor mutation is actually associated with obesity and other diseases that's associated with glucose metabolism.
So that's the lack of melatonin receptors, you mean?
Yes, I think what the body was trying to do is to increase the melatonin receptors so that it actually gets more melatonin because of decreased production. But while the body is evolving and doing these genetic adaptations, you know that it's all experiments, right, in the past. If something works, then you have positive selection and the gene stays. And nowadays, this positive selection doesn't really work anymore because nobody dies anymore. So you end up with people with these melatonin receptor mutations. And now there are different types of mutations. Some of these mutations will actually elevate and shift the melatonin production cycle. And they, instead of stopping endogenous production at 8 a.m., which is normal for a lot of people, they continue producing melatonin up until like 10 a.m. or even 11 a.m. at high levels. And the thing with melatonin is it suppresses insulin so that it raises blood glucose. That's a protective measure. At night, when you don't eat, in the past, when there's no food at night, your blood glucose drops and you have to maintain the level. And that's what melatonin does. It helps you maintain high blood glucose at night when you're sleeping, when there's no food. But nowadays, people eat at all sorts of hours. They eat at night. There's no such thing anymore. And so this ability of melatonin to suppress insulin during the daytime is actually quite a problem for people with these mutations. Because if they eat a big breakfast at 8 a.m. while melatonin production is still high, you can end up with a lot of glucose issues.
Wow.
So, going back to what you said about stopping melatonin production, you don't just going outside in bright sunlight.
But it was the color. The color of the light was like.
Do you remember what color the light was?
Yes, it was like a greenish blue.
Okay, blue light. Blue light reduces. Now, I'm really, really glad you mentioned blue light, because a lot of people think blue light inhibits melatonin production. That is only partially true. Blue light in the daytime inhibits melatonin production, and at the nighttime, it also inhibits melatonin production. But blue light in the daytime can also increase melatonin production at night.
Okay.
So that's probably why you fell asleep, because, because what it is there were quite a few experiments involving humans and mice. With the mice, they put the mice in different color cages, and they found that the ones kept in blue cages had the most melatonin at night. And in humans that had cataract operations that use the lenses that filtered out blue light.
Blue blockers.
Blue blockers, yes, inside the lens. They actually had lower melatonin at night. If you filter out, if you completely have no blue light, you end up with less melatonin. So what I think happens is, if you are exposed to blue light in the daytime, blue light increases oxidative stress. And it is actually a natural response for the body to say, okay, I've got all this oxidative stress. I've got to increase my melatonin production at night to work on fixing all these oxidative damages. That's what I think happens. And so that's why more blue light in the daytime can increase melatonin at night.
Okay.
We have a question. Somebody says, if I can't get intravenous vitamin C, can you get enough ascorbic acid orally? Is it best to take it on an empty stomach or with food, and how much to take?
I've actually covered this in depth in a few presentations before. A lot of people are under the impression that oral ascorbic acid does not reach the same plasma concentration as IVC, and that is not correct, because all the previous studies that examined plasma levels, they examined it at a period of usually 1 or 2 hours after the oral intake or the IVC injection. A study that tracked plasma levels minute by minute compared oral vitamin C to IVC, found that oral vitamin C actually had I would say at the peak, the initial peak, 10-fold magnitude over IVC. And that's because the oral oral ascorbic acid is transported by the high capacity SVCT1, whereas IVC, which is sodium ascorbate, is transported by the low capacity SVCT2. And I think if you want a detailed understanding of the difference between the oral and the IVC, go to the first link I provided after the summary. That would explain that in full detail. And all the slides I've presented today will be uploaded at my blog, and I will give you a link so that you can share with all the audience today.
Yes, and we have it as well.
Yes.
Steve, you asked about the ascorbic acid supplementation on a daily basis.
Yes. Are you asking how much
Yes.
you should take? Okay.
And how
Now
to take it.
Huh?
Yes,
How to take it.
How to take it? Okay.
And also how much?
How much? First, how much? That depends on the person, because every person has different redox requirements. A person your age, similar background, similar health challenges, may require a different amount. So you actually have to find your needs by testing to bowel tolerance, which is taking ascorbic acid of, let's say, 1 gram an hour until you start to get loose stools. That is a sign that you've reached saturation. And some people may reach saturation after 2 grams, some people 10 grams, and I've had people have no reaction after 35 grams. So there is actually no guideline as to what is the most optimal amount for anyone. But in general, I would recommend for any adult in a big city with high EMF to have at least 6 grams or 6,000 milligrams of ascorbic acid every day, taken in 1 gram doses. And with COVID, I have increased that amount to between 10 to 12 grams. And if you are a healthcare worker in a high risk environment, you must take ascorbic acid at least 1 gram an hour, 1 gram an hour. And you can take it either in capsules, tablets, or powder form. And if you have, if you reach bowel tolerance and if you need to take more, that is when you're sick. But usually when you're sick, your bowel tolerance will be increased exponentially. Let's say normally you get loose stools at 8 grams. If you're sick, you can go to 20, 30 grams without having any effect. So that's also one way to tell if you're sick.
We also have a question here about how to take melatonin, whether sublingual or oral, and have a personal question about, I use liposomal melatonin, which is the only form of melatonin that gives me a hypnotic effect. And I was wondering if you could speculate as to why.
So you've tried other types that doesn't, at the same dosage,
Yes. Well, actually, liposomal gives me a hypnotic effect at 1/6 the dose that I can tolerate with the oral or sublingual or sustained release.
Ah, okay. Let me speculate on that. In the gut, we have a lot of bacteria, and they all love melatonin. So you may have a lot of friendly or unfriendly guests in your gut that quickly takes up the melatonin before it reaches your liver. But the liposomal form could protect the melatonin and delivers it to your liver for it to be metabolized. So that is one theory that is possible. And the original question is sublingual or oral. Sublingual is very effective. And but in general, most people, if they don't have issues with metabolism, oral form is fine, and they really don't need timed release. Because, I'll tell you why. Melatonin reaches the blood very quickly, and it starts to get metabolized. But the thing is, the different metabolic forms are still very effective as antioxidants. So it just keeps breaking down and your body can still use it. So I'm not too concerned. But if you're sick, you need a constant supply. And that's why when you have COVID, I suggest even in the daytime to take a small amount of 1 to 2 milligrams per hour. And at night, take a mega dose depending on the severity. And probably if you are going to the my highest recommendation at night, which is 18 milligrams, to divide it into 2 doses of 40 milligrams at night.
Okay, we have 2 questions here about, is it a good idea to increase your melatonin on days when you're going to be doing heavy exercise?
On the day, before or after? Did they
Well,
specify?
they don't say, so both.
Okay. Okay. Now, as we can see, melatonin has an immediate effect of increasing OXPHOS. So if you're exercising, I think taking it before. Now, if you are exercising and not for dealing with infections, you really don't need that high an amount. Physiological dose is only 300 micrograms for an adult. So you for enhancing exercise, take 100 micrograms. That's all you need. And if you overdo it, you might, some people say they get sleepy, but I myself use melatonin in the daytime and I never get sleepy from it. But I would suggest to the person, try with 100 micrograms, and see if you get any effect, and then increase it, because everybody's reaction is different. There is no, how your mitochondria reacts to exogenous melatonin would be different. So find your own levels by experimenting.
And what about after, if you've done heavy exercise, for recovery afterwards?
Then take some more melatonin that night. That's when you regenerate. Take the melatonin at night.
And would it be worth taking over and above physiological dose then, or?
Well, depending on how stressed you were, and and what type of exercise, and now that's assuming you do not regularly take a physiological dose. Is that correct?
I've been experimenting from physiological and a bit further up. I'm 54. I've had cancer. I've had high ferritin levels. So I've been following your blog for a while, but I've just experimented with dosage up to 10 milligrams, 10 milligrams after like a heavy squash game.
Right.
Helped me sleep because I couldn't normally sleep after an evening squash game.
Right.
With that for starters.
Yes. So if 10 milligrams help you, I would stay at that level because I think that's a good dose, and see how well you recover the next day, and then work from there.
Okay. All right. Thank you.
You're welcome. And congratulations.
Yeah, we have another question here about whether or not melatonin interacts with the cortisol rise that takes place at night.
Now that is actually a good question. There is a mutually inhibitory effect of melatonin and cortisol. Now you would have to ask, what are the conditions that increase the cortisol? Usually if you take melatonin, it will suppress the cortisol. But it depends on what were the conditions that raised the cortisol to begin with. Do you understand my question?
How about low cortisol in the morning is the symptom that the person experiences? Does that give you a context?
This is Levy. I asked the question. Yeah, my curves are off because of adrenal fatigue. So high cortisol at night, extra low cortisol in the morning. That's the cause.
Okay, adrenal fatigue. Yes, adrenal fatigue, you need ascorbic acid and melatonin. And over time, that will rebalance your cortisol.
Wow, we've really gotten to the end of this?
Yes, good.
Susan, do you have any questions that have accumulated?
No, but I just want to thank you for that very thoughtful, insightful talk, and I want to run out and get some melatonin.
I have a question. One more question?
How about Muriel?
Yes, can you talk a bit about how much natural variation there is in natural melatonin between different individuals?
Of the same age?
Yes.
Male and female?
Well, I'm a girl, but I think everybody might want to know.
Okay, you're asking me, you have to tell me what age.
Well,
you're thinking of.
I'm 66.
Oh, 6-year-old, a 6-year-old.
Well,
No, 66.
66. Okay, sorry.
Thank you.
So you're asking me, what is the range of endogenous melatonin in a 66-year-old female, and what can affect the production levels? Is that correct?
Because I feel quite energetic most of the time, and I'm in good shape, and I, wake easily, I sleep easily. So does that indicate, anything?
Okay. If you sleep well and if you have energy, usually that would indicate you have a fairly decent amount of melatonin. And what is interesting, and that I found that is, if you have trouble sleeping, taking melatonin may not help. So melatonin, to be called a sleep hormone, is actually not quite an accurate description, because you cannot have good sleep without melatonin, but you may not have good sleep with melatonin. And if you have good sleep, usually you have enough melatonin. And if you are wondering if you should take melatonin because you sleep well and you are functioning well and you have energy, I would suggest that you experiment with just maybe 100 micrograms of melatonin at night and see if you sleep even better or have even more energy during the daytime. And if that does help you, then you can usually, you would benefit by melatonin, because at around 66 years old, most people do have lower melatonin levels. And if your weight is normal and you are not, you don't have any metabolic issues, then you probably do not have any melatonin receptor mutations that would make you have a higher level of melatonin production. And by taking a physiological dose of just 100 micrograms regularly at 66, I can almost say that you should look forward to a very healthy aging process, because melatonin also is neuroprotective, and it has been found to be protective for neurodegenerative diseases like Alzheimer's and Parkinson's. So there is absolutely no reason not to take a tiny amount, even if you sleep well. That is my suggestion for you.
Plus, it can protect me against COVID. Thank you very much.
Oh, you're welcome. Thank you for being here with us today.
I have a question.
Yes?
Can I ask it?
Yes.
In something that I read a lot of Doris's stuff, your stuff, Doris, and somewhere I read a little hint that transdermal melatonin was looking really promising to you. So I'm wondering, A, how can I find it, or can I buy the powder and mix it into some kind of a cream or lotion that my body is okay with?
Yes. In fact, I was working on a book called Beyond Longevity, which features melatonin and ascorbic acid. And in that book, I was going to go into great lengths about how to use melatonin in the most optimal fashion. But then COVID-19 happened, and I decided, well, at first I decided I wasn't going to talk about melatonin because most people have really very biased opinions about exogenous melatonin supplementation. And I thought that, well, I'm just not going to talk about it much, because just talking about ascorbic acid took a long time to get people to understand that it is very important to take ascorbic acid. So I was just focusing on writing the book. And then COVID-19 happened, and I realized, wow, melatonin can be really, really useful against this virus. So I started to write about melatonin. And COVID-19 actually helped put melatonin in the limelight and position it should be. And everybody is now understanding the importance of melatonin supplementation, which is excellent, because I have a feeling that everyone will end up with healthier bodies going forward, because they are taking a natural molecule, ancient molecule that's been severely deficient because of our lifestyle of high light at night. I mean, people are basically deficient in melatonin because of bright light at night. So going back to your question, yes, I have proprietary formulas on how to use melatonin transdermally, and I have to say I'm going to be very selfish and not share them now until I release my book. Is that okay?
Yes, I have a question here. Do you know of any reason that you would want to have melatonin tested in some way, blood test, urine test, or whatever? Or do you think that the best way to test it is by actually taking it and seeing what happens?
Now, it depends. If you suspect you have melatonin receptor mutations, you should take a 24-hour saliva test, because that way you can tell if your phase are out of sync. And usually 24-hour melatonin saliva tests are taken at 3 points during the daytime. I would suggest 8 a.m., 3 p.m., and 3 a.m. Usually people with receptor mutations, they have very high 8 a.m. levels, higher than the 3 a.m. 3 a.m. is supposed to be the peak, but in those people, they don't have that peak. And so for those people to understand when they have natural peak production is very important. So yes, but for other people without any obvious metabolic issues, I don't think it is necessary to test for melatonin levels. And the best way to find out how much melatonin you require is by experimentation.
Are there any ways to measure SNPs relating to the receptor mutations?
To measure what receptor?
Well, the single nucleotide polymorphism.
Oh, the SNPs. Oh,
SNPs.
yes. Yes. If you've done a 23andMe test, and you go into the website and go under, look at your raw data information, and just type in MTRN1B, it will list all your SNPs related to that gene. And that's how you find out your SNPs. But there are many SNPs related to the melatonin 1B receptor mutation. There are two major receptors, 1A and 1B. And the 1B receptor is the one that's associated with diabetes, obesity, and other metabolic issues. Now, different SNPs and different ethnicities have different effects. So it is actually not a straightforward thing, and there is right now still no consensus on the SNPs for the mutations.
Okay, I think we're down to our last question. Sheila?
Hi, Doris.
Hi.
So my question is a little bit roundabout. I used to be a severe night owl, and I mean, up reading in LED lights in front of computers 2, 3 in the morning and not really sleepy. And I knew this was not good for my health. And I was trying to find a way to reset my circadian rhythm.
Mm-hmm.
And at that time, melatonin really had no effect on me. It didn't matter when I took it and what dose I took. But I started fasting. And after I would do 60-hour fasts, I noticed that my circadian rhythm had changed. And when I went back to incorporating a very small amount of melatonin, it seems that I'm now, and I try to be in bed at around 10 or 11 because I've read that peak production typically is around midnight.
3 a.m.
Oh, 3 a.m. Okay, well, that's good to know. So
yes.
What is happening that a fast would reset the circadian rhythm. And like now I'm like, my body is telling me to go to bed at 10 o'clock. And as soon as I get that melatonin in me, like I really just want to hit the pillow now. And that's like a complete switch has been tripped.
That's actually truly amazing that you experienced that. Now, fasts or even intermittent fasting or calorie restriction all have effects on pathways that affect metabolism and even longevity. And when you fast for how many hours did you fast? And was it a water fast or what type of fast was it?
I just did 60 hours, and yeah, I just drank water. If I had like severe hunger, I might make some heat up some bone broth, but that was it. No juice, no glucose.
When you deprive your cells from nutrients, a lot of pathways are activated. And I don't recall specifically that fast actually resets circadian rhythms, but I am not surprised that it does. In fact, one of the things that affects regeneration and longevity is specifically the limitation of nutrients, because what happens is your body, in order to survive, they all of a sudden activate all sorts of pathways to make sure that the sudden reduction of nutrients do not start to affect the organism. And the proper use of fasting can increase longevity. And in your case, it actually reset your circadian rhythm. So that is, I must congratulate you for being able to do that.
Well, thank you. It was a side effect that I wasn't quite expecting, but I mean, I'm familiar with the autophagy process and that, you know, that cleanup process that's happening. But I'm quite blown away by it. I literally, my body is saying, go to bed. I'm like, I'm about to fall asleep. It's quite nice.
That's amazing. That is amazing. Yes.
Susie?
Oh yeah, just one more. You were going to talk about the MFTHR. Yeah, so just want to bring that up again.
Was that a question?
Yeah, you were going to say something about the MTHFR SNP, I think.
Oh, the melatonin SNP.
Oh, the melatonin SNP.
Yeah, so I'm the one who asked that question again. MTHFR is perfect. That was in relation to COVID and the heart issue, because the MTHFR creates propensity towards clotting, blood clotting. So, yeah.
You need to repeat that, because there was some echo and your voice broke up and I couldn't hear you.
Sure. MTHFR, which which creates a tendency towards blood clotting, and then the heart things that we talked about related to COVID.
Okay, that mutation actually involves a lot of pathways, and in COVID and the clotting issues, they are interrelated, and so, okay, I need to understand which aspect of the mutation at its effect on clotting, is that the question?
Yeah, I've noticed its effect on clotting very severely, and recently, after a traumatic brain injury, actually, that manifested.
So you had hypercoagulation, is that what it is?
Yes, exactly.
Okay. Now, the hypercoagulation can be related to platelets, and also the now I have to start talking about the CD147 aspect, which affects the genetic expression of von Willebrand factor, which affects clotting.
That's a whole topic unto itself.
Yes, I know, because I spent like 2 hours talking about that. Now, in my paper, I actually talked about the whole pathway of the clotting. And so you might want to refer to that to understand the clotting aspect. But as remedies, anti-clotting, melatonin, again, and ascorbic acid. So how much ascorbic acid and melatonin are you taking, Susan? Susie?
Levy.
I'm taking none of either.
None.
Yes.
Okay, so you might want to start to take some, because
Yes.
Yes, because that will help your hypercoagulation.
In the same way that you recommended throughout this, to titrate
Yes.
and test. Okay. Okay.
Yes, and if you have an active infection showing other symptoms, you should follow the way I have described the dosage in the day 1 to 4 protocols.
I will. Thank you so very much.
You're welcome.
Please come back.
Yes, this has been a fantastic presentation. We're at the end of the questions, and so I think it's probably a good time to give you time to work on your next paper and maybe even take a nap. So,
I think taking a nap will be good.
thank you so much for being here.
You're most welcome. It's been a pleasure, and it was so much fun. Thank you, Susan, and thank you, Steve.
Thank you.
Thank you.
You're welcome.
Fans love you.
Thank you. Thank you.
Bye-bye.
Thank you.
Bye.
Bye.
Hi.
Bye. Hi.
Hi, Doris.
I didn't think you'd be here.
I'm here.
Okay, cool.